I don't think so, so that's good.
Okay, says we're live. Okay, great. Well, hi everyone. This is lesson 9, and I'm Heather Benson.
And I'm Jessie Packard.
And here we go. I will explain question number 1 here, and I'm going to turn on my screen share so that you can see what I'm saying. Okay, we see that okay? Okay, great. So for number 1, it asks about drawing a diagram of an intermediate stage of DNA replication in the bacterium E. coli. And so it asks to, make this full screen, it asks to include several different things. And so I'll just go through that. So we have the DNA replication happening here. And so the first thing that it wants to know is the ORIC, and that is over here. And then after that, it wants to know the DNA primase. And DNA primase is over here on the lagging strand. And then we have DNA helicase, which is over here on the end. And that there's actually a DNA helicase over here on the opposite side. And then RNA primers for the strands. So we have an RNA primer right here going that way. And then 2 leading strands. So we have a leading strand here and a leading strand up here. And it kind of is sort of a mirror image going diagonally there. And then we have DNA polymerase 1, and it's all the way over here, that little guy. And then the next thing is DNA polymerase 3 alpha subunit. And make sure I have that right there. So it's right here, and it's with the alpha subunit. And then it wants you to include several Okazaki fragments, and they're right here on the lagging strand. So yeah, let me go over here.
Awesome.
Turn off my screen share. Okay.
Perfect. So for question 2, this was just fill in the blank. So for the protein complex that synthesizes primers is going to be primase. It's got it right in the title, so that one's pretty easy. The protein that promotes separation of the AT-rich sequence with the ORIC, that's going to be your DNA A. And the polymerase 3 ellipsum subunit is the part that does proofreading and that part of the synthesis. Polymerase delta is the protein synthesis that does the leading strand for eukaryotes. And the TUS or TUS protein displaces the DNA polymerase 3, and that's used for the termination during the replication process. And for F, polymerase 1 is the protein that does the 5' to 3' polymerase and exonuclease activity. For G, topoisomerase 1, this induces the negative supercoils and separates the force. The ORC binds directly to the ARS elements. The SSBPs, or let's see, I've got the long name here. The single-stranded DNA binding proteins. Those prevent the DNA strands from going back together and sticking. And for J, it's going to be DNA ligase that seals the Okazaki fragments together to create one continuous strand of DNA.
Cool. Okay, I'm going to turn this on again for question number 3. Okay. So for question number 3, it asks about the enzymes involved in DNA replication. And part A asks to list 5 enzymes that consume ATP during DNA replication, and they can be prokaryotic or eukaryotic. So we have DNA primase, DNA gyrase, DNA helicase, DNA A protein, and DNA ligase. And then part B, it asks to list 5 enzymes again that consume DATP during DNA replication. And there we have alpha, delta, epsilon, and DNA polymerase 1 and DNA polymerase 3. And part C lists 5 enzymes that have the 3' to 5' exonuclease activity. And those are actually exactly the same ones up here. So all of these are also the same answers for part C. For part D, indicate one enzyme involved in replication that uses DNA as a template to synthesize RNA, and that is DNA primase. And then part E is indicate one enzyme, prokaryotic or eukaryotic, that uses RNA as a template to synthesize DNA, and that would be telomerase that uses RNA as a template to synthesize the DNA.
Awesome. So for question 4, you are synthesizing a DNA strand. And so what I found was helpful, I did the complementary strand for the part that they give you. And so for the nucleotides, that's just going to produce a normal strand of DNA. And so going from the 5' to 3' end, you should have A, C, T, G, A, T, G, G, T, C, C, T, G, C, T, A, T. So that's just a normal strand. There's nothing wrong with that one. For part B, because you have the DCMP, the C nucleotides from being synthesized. So when you go backwards, starting from 5' to 3', you are only going to end up with an A because the next one is a C and that doesn't get synthesized. So, and then for part C, because you have the UTP, that doesn't allow the T nucleotides to be synthesized. So when you go along your strand, you're only going to go A, C, and because there's a T for the third one, you can't go any further. So you're just going to have an A and a C for that part. And for part D, because you have the GTP, your G nucleotides aren't going to be synthesized. So the furthest you can get is from the 5', it goes ACT, going to the 3' end, and that's as far as you can get.
Okay. So for number 5, it gives you a long list of proteins involved in DNA replication, and it asks a few different questions. The first question for part A asks which of the above proteins is found in prokaryotes, and there's a bunch of them. So we have DNA gyrase, TUS, DNA A, DNA primase, DNA polymerase 1, and DNA polymerase alpha subunit. For part B, which of the above proteins is found in the mouse musculus, and that is eukaryotic. So what we have there is CDC6, DNA polymerase epsilon, DNA primase, MCM helicase, DNA polymerase alpha, telomerase, and pCNA. For part C, which of the above proteins is involved in chromosome compaction? So here we are talking about termination. And so for there we have telomerase, and I think that might also be the TUS. I've meant to include that there. And for part D, which of the above proteins is involved in the initiation stage of DNA replication? And that would be CDC6, DNA A, and MCM helicase. For part E, which of the above proteins is involved in the termination stage of DNA replication? And we have DNA gyrase, TUS, and telomerase. And yeah, that's about it for all the problems there.
That's it.
So, hope you enjoyed watching.